Biological discovery is a process of building on earlier achievements. Several important steps led to the discovery that DNA holds the hereditary information. Let's look at how biologists first determined that chromosomes are the mediators of inherited traits. In the late 1800s, Gregor Mendel, an Austrian monk doing experiments with pea plants, discovered that specific traits are determined by inherited factors, later called genes. Mendel's work went largely unnoticed until the early 1900s, when American biologist Walter Sutton and German embryologist Theodore Boveri independently noticed that the behavior of chromosomes in egg and sperm development exactly matched Mendel's theory of inherited genes. This led them to believe that Mendel's genes are located on chromosomes. They proposed the chromosome theory of inheritance, which stated that chromosomes carry genes, the hereditary information. The chromosomal theory of inheritance was further supported when Thomas Morgan was able to figure out that the gene that gave fruit flies red eyes was. The next groundbreaking experiment in the discovery of DNA as the genetic material was the first bacterial transformation, performed by British medical officer Frederick Griffith in 1928. Bacterial transformation is the process by which bacteria are changed by the uptake of foreign DNA. But at the time of Griffith's experiment, nothing was known about bacterial transformation. Griffith was studying the bacterium Streptococcus pneumoniae, which Griffith found that the smooth cells were able to kill injected mice, while the rough cells could not. When he used heat to kill the smooth cells, they were no longer able to kill the mice. However, when heat-killed smooth cells were mixed with the non-lethal live rough cells, the injected mice died. And live smooth bacteria could be recovered from the dead mice. Somehow, the rough cells had acquired the ability to make the polysaccharide coat and were now able to kill the mice. At the time, Griffith didn't know what made the rough cells become smooth cells. However, his experiment gave other scientists a starting point to look for the nature of the genetic material responsible for the In 1944, American bacteriologists Oswald Avery and MacLeod used the data from Griffith's experiments to determine what the transforming agent was. They purified various substances from bacterial cells and tried to transform rough bacterial cells with each of the substances individually. Avery and MacLeod found that only DNA was able to transform the bacteria and make the bacteria deadly to the injected mice. The rough cells picked up the smooth cell DNA and began making the polysaccharide. In 1952, American biologists Alfred Hershey and Martha Chase devised an experiment to determine if the genetic material in a bacteriophage is protein or DNA. A bacteriophage, or just phage for short, is a virus that infects bacteria. Viruses are much simpler than cells and are composed of DNA enclosed in a protein coat. Scientists hypothesized that the genetic material of the bacteriophage must be injected into a bacterial cell during an infection. Hershey and Chase used radioactive isotopes to keep track of the locations of the protein coat and the DNA during an infection. A radioactive isotope is an atomic form of a chemical element that is unstable. To reach a more stable state, the isotope gives off a detectable particle. Hershey and Chase incorporated radioactive isotopes into the bacteriophage. Proteins contain sulfur, but little phosphorus. Hershey and Chase incorporated the radioactive isotopes separately into 2 samples of bacteriophage and then used the radioactive bacteriophage to infect bacteria. They placed the infected bacteria and bacteriophage mixture in a blender and agitated it. Only bacteriophage material that was injected into the bacteria would remain in the cells. Hershey and Chase separated the bacterial cells from the medium in which they were growing. Bacteriophages inject only their genetic material into bacteria, so bacteriophage DNA, but not protein, was found in the cells. Bacteriophage DNA, marked with radioactive phosphorus, entered the bacterial cells. The protein coat, marked with radioactive sulfur, remained outside the bacteria and was found in the medium.